Now we move to our last speaker, Dr. Rohimah Mohamud. Hello, Dr. Rohimah Mohamud. Can you hear me?
Hello. Assalamualaikum. Yes, very clearly.
Hello.
Hello.
Yeah, Waalaikumsalam. How are you, Prof?
I'm good. Thank you. I'm good. Thank you. Yourself?
Okay. Yeah, fine. Okay. Okay, ladies and gentlemen, before we start, I will give brief biography of Dr. Rohimah Mohamud. She is currently a lecturer at School of Medical Sciences, Health Campus, University Science, Malaysia. She completed her master degree at USM, Malaysia in vaccinology and continued her Doctor of Philosophy in Immunology at Monash University, Victoria, Australia. Now she will talk about update on COVID-19 innovation, including detection and vaccine. The following speech will be discussed about the effect of COVID-19 pandemic to many health areas, including the recent development of COVID-19 detection technique and vaccine, and also challenge how to promote healthy life and well-being in the after pandemic era. It was interesting. Please welcome Dr. Rohimah Mohamud. Time is yours, Doctor.
Okay, thank you very much. I just want to share the screen. Okay, we go to this share screen.
Okay.
How can I do that? Because it says one participant can share at one time.
Okay, please. Wait, Doctor. I will check.
All right.
Is it okay to share?
Can I share now?
Yeah, okay. Please, Doctor.
Okay, when I click share screen, is it advanced sharing options?
Click the bottom of Zoom, share screen, Doctor.
Yes, yeah.
Share screen.
Yes, but okay, yes,
Cannot?
yes, I can see that.
The bottom of the Zoom.
Okay.
Okay, it's okay.
Yes. Okay, share.
Yeah. Finally. Yes. Yes, Doctor.
Okay, shall I proceed?
Please.
Okay.
Clearly.
Okay, thank you. Assalamualaikum and hello everyone. First of all, I would like to welcome everyone to this Asian Academic Society International Conference. And I would like to thank and congratulate all the key speakers that has already delivered their excellent talk. So my name is Rahimah binti Mahamud from the Department of Immunology, University Science Malaysia. And today I'm going to give a talk on update on COVID-19 innovation and innovation detection and vaccine development. Okay, inshallah. So this is the outline of the talk. So current research that leading by my group on pandemic to the health areas. For this one, a little bit briefing only. Refer to the recent development of COVID-19. You give me only 15 minutes. I think that's very short. Okay, as an introduction to my research group, I divided the research into the basic and fundamental research. And this is what I'm going to focus on the development of nanomedicine. For this one, we focus on the therapeutics as well as on the diagnostic. And okay, For the fundamental research, we intend to know why, what, and how tumor necrosis factor 2, that is TNFR2, mediate our immune responses towards these diseases. And we are also investigating what are the effects of all of these nanoparticles on the TNF-R2 cells and how they can regulate our immune system. Because I'm at the immunology department, it gives an effect. So that is what we are focusing on. And as you can see on the screen, there are different types of polystyrene nanoparticles, silica, iron oxide, and etc. So when all of these nanoparticles goes inside our body, the macrophages, dendritic cells, it has the different effect depending on the different characterization of nanoparticles. Okay, I give one example. If we use big particles such as like 0.5 micron particles, so it will target the minds. But let's say if we use 50 nanometer particles, or we call that nanoparticles, so it will target the dendritic cells. So we know that in vaccine development, dendritic cells are important cells. That is the bridging between innate immune response as well as adaptive immune response. So these are the what happened in our lab basically. Okay, so my group, we just actually received or secured by a funding by Asian University Network or SIDNet JICA for the special program for research for COVID-19, that is we call SPRAC, and the project on the COVID-19 is on the ultra-sensitive compact immunoassay system expressed biochecker using fluorescent Janus particles for the rapid detection of SARS-CoV-2. So for this one, we have the collaboration with Tohoku University, and the project started this month. Actually, no, I mean last month. So they will post their nanoparticles to
Sorry for the inconvenience technical. We will try to connect to the Dr. Rohima. Just a moment. Hello, Dr. Rohima. Now we share screen for our slides.
Hello.
Hello, welcome back, Dr. Rohima.
Sorry for that. Shall I proceed?
It's okay.
Where am I? Is this at this
Yeah, we lost
slide?
connection for a while. This is slide
So
from
this slide or the other slide?
committee, right? Yeah.
Okay, that's good. That's good. Shall I continue
Yeah,
on this
yeah.
one?
Okay.
Okay. Okay. Now we just go through this slide. Okay. Now because is it clear?
Now clear.
All right. Now we
clear.
focus on because okay, we expected to get this the particle next year. Next slide. Okay, this is the effects of COVID-19 pandemic to health areas. I'm not going to go into details. We may refer to this report. And 2016 was considered as the, what do you call, start of year for SDG after 2015 MDG data, which published in 2017. So the report present the data for Asian and also for Asian member states. So the data were gathered and compiled by the national as well as national statistical officers in the Asia region from 2016 to 2018. And next slide, please. Um, Okay, this is the page 5 of the SDG report. We can see that Asian and Asian member states are facing issues and challenging in the what we call collecting the data and etc. And these problems are worsened by the COVID-19 pandemic. Okay, we just go through. Okay, in spite of the issues and challenges based on the SDG report that I show you, we are working hard to keep everyone going forward in achieving the sustainable nature. And besides that, there is a well-established health system support by other countries as showing in this slide. Can we go to the next slide? Okay, I'm not going to talk more on the effects of health post-COVID-19. So now we go to the virus detection. And here you can see that there are different type of virus detection technique. We can see that PCR, RT-PCR, we have the TN. Okay, next slide, please. Okay, for the detection standard, okay, the standard 19 are the molecular and serological method, as you can see here. So these two methods are the main method to detect the virus. So if the individual get the positive, they have to do first they get the swab and then they perform the molecular methods. And second, they take the blood and do all the serological methods. So in the next slide, I'm showing. Okay, these are the comparison of the molecular methods based on the sample, accuracy, time, professional skills, need, POCT, availability, cost, and high throughput. So we can see here the differences between these three. And all of these three methods use RNA sample. And in terms of the accuracy, all of these methods have high accuracy. Okay, for the times, we can see that RT-PCR, it takes more time compared to others. And all of these methods are limited in the term of availability. And the price are, yes, expensive. And then we can see the high throughput. That is the use of automated equipment to rapidly test thousands to millions of samples. And this is very important to test millions of COVID-19 samples, especially by using the RT-PCR. Okay, remember we have the what we call molecular methods, and another one is serological methods. So this one, we have the lateral flow assay and ELISA. Okay, for the samples, all of this using the blood, so it's more to detection of antibody or antigen, and then for accuracy, it is from low to moderate, and the cost is a little bit lower compared to the molecular methods. Okay, so for next slide, please. So for the detection SARS-CoV at the early infectious stage by a rapid and accurate diagnostic method, that is important to save thousands of lives. So now I'm going to the other point of my talk. So this is the important thing. Go to the next slide, please. So which is on the vaccine development. Okay, currently there are 9 vaccine candidates in phase 3 of the clinical evaluations, and with the different vaccine platforms. And I'm going to show you there are 5 vaccine platforms. So here we can see that this is what we call viral vector vaccine. So as you can see on the top of the slide, that is number 1, viral vector vaccine. So it involves, maybe it can be like to carry the gene. So most of the gene from the SARS-CoV, they focus on the spike protein. So the gene from the spike protein is inserted into the viral vector, and this what we call candidate vaccine will be inserted into the cells, and you can see the other processes. Okay, next slide, please. So this is another platform. We call that DNA vaccine. So as you can see here, the number 2 over there, DNA vaccine. It involves then A that encodes the target antigens. So as I mentioned, the surface protein gene inserted into the plasmid. So you can see that the plasmid bring or carry the specific genes for the SARS-CoV surface protein, and then goes into the cells. So basically, people do electroporation and etc. All right. Um, next platform is the RNA vaccine. So as you can see there, number 3, please. The messenger RNA or mRNA encoding SARS-CoV surface protein. So just now we have then a vaccine that using the plasmid, but this one is they use RNA. Yeah, because RNA just insert into the cells, they can basically self-amplify inside the cells in some of the RNA vaccine system. All right. So,
Excuse me, Prof.
okay, I forgot to mention.
1 minute left.
Pardon?
Sorry, you still have.
1 minute left.
Okay.
Okay. The last one. Okay, go to number 4, please. That is the live attenuated vaccine. This one is attenuated, live attenuated or kill vaccine. People do kill either by the chemicals or irradiation. And then last one is the protein-based vaccine. So this is like a small protein from this SARS-CoV, and then basically people target or couple with the nanotechnology platform. So all of this vaccine is to evoke the immune system. Okay, so we've gone through all 5 different vaccine platforms. Okay, can we go to next slide? Next slide, please. With the table, next slide, another table. Okay, and then you just click. Okay, as I mentioned, we have like different vaccine platform. We have inactivate on vaccine. We have the what we call recombinant viral vaccine. You just click, that's fine. And then we have the protein-based vaccine. So all of these 9 vaccine that is already at the clinical phase. I mean, phase 3 clinical evaluation that's ready to be what we call commercialized. Okay, these are the example. I list down all 9 vaccine. You just can keep going. Just keep going. Okay, yes. Okay, that's one. Okay, yep. All 9 vaccines. So you can refer this to the clinical trial. Okay, this is the conclusion. What is the direction for the future research? Ongoing progress of candidate vaccine through the preclinical and clinical studies. Yeah, this is a must. And ongoing detailed characterization of the immunopathologies of the COVID-19, implementation of large-scale post-marketing surveillance system to monitor SARS-CoV vaccine safety. Okay, and lastly, strengthen the vaccine development using a pandemic paradigm with the overlapping phases to shorten the development time. So because we have the pandemic and then the vaccine development, it cannot be using the traditional way. It has to be the pandemic paradigm. It means that it is a very shorter time. Okay, so lastly, I would end my presentation. Okay, before I end my presentation, I pray that all of us to stay healthy, practice social distancing, wear face masks when possible, and avoid crowded areas as the preventive measures against the COVID-19 coronavirus. So with that, I thank you for your attention, and I welcome any new collaborator to our group. With that, I thank you.
Thank you very much, Dr. Rohima Mohammed, for this, the presentation. It was interesting topics that we're still waiting for the availability of vaccine.